Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this particular module, we are discussing about the cell growth and the cell division. So far what we have discussed about the importance of the growth and how the growth is being regulated within the prokaryotic system or the eukaryotic system. In this context, we have discussed about the cell checkpoints, how the cell growth is being regulated by following the different types of stages, and each stage within the cell growth is going to be regulated by cellular machinery or the set of proteins. Subsequent to that, in the previous lecture, we have also discussed about the different types of events, what is happening with the within the cell growth, and how you can be able to provide the nutrition into the cellular system. So we have discussed about the preparation of the prokaryotic media for the prokaryotic system, and then we also discussed about the media for the eukaryotic system. Subsequent to that, we have also discussed about the different types of method for monitoring the growth of the prokaryotic cell and as well as the eukaryotic cell. Within the eukaryotic cell, we have discussed about the monitoring of the growth for the animal cell and as well as for the plant cells. So now it is clear that growth is very important for the longevity and as well as the survival of a particular the living organisms, and the growth is being divided into the different stages, right? And all these stages are part of the cell cycle, right? So when a cell is growing through the different stages of the cell cycle, it actually follows a very regulated manner, and that's how it is actually going to be accomplish the purpose of cell division in such a way that it is not going to give rise to the abnormal cells, or it will not give rise to any kind of abnormality. So when you talk about the cell cycle is been in the eukaryotic system. The cell cycle, the cells undergo the different stages, and these different stages are being regulated by the cyclins and CDKs and all those different molecules what we have discussed in this particular course. So in today's lecture, we are going to discuss about the cell cycle and how the cell cycle is being, can be studied within the biological system, and how you can be able to study the different stages of the cell cycle. So cell cycle is, as we say, right? Cell cycle is being done in the eukaryotic system. The eukaryotic cell undergoes the precise cell cycle and division to produce the two daughter cells. Cell cycle is a series of tightly regulated events leading to the division and the duplication. It is a vital process used for the single cell fertilized egg to develop into full organisms. Cell cycle is the crucial event underlining the regeneration and repair in tissues, liver and heart. This is very important, and this part I think we have discussed when we were discussing about the first lecture, right? That why the cell growth is required. The cell growth is required so that you can be able to replenish the damaged cells, and also it will help you to bring the repairing process, right? In prokaryotic system, the parent cell is dividing simply by the binary fission, right? And that's why the cell cycle is not that relevant in terms of the prokaryotic system. But even then, we have discussed that in the prokaryotic system also you have the different stages. Stages like you are going to have the growth, cell growth, then you are going to have the cell synthesis of the DNA, and then you are going to have the division, right? Whereas in the eukaryotic cell, the cell has three different stages. Interphase, where you are going to have the synthesis of the genomic DNA and the cytoplasm. Then you have the mitotic phase, where you are going to have the division of DNA into the two halves, and then you are going to have the cytokinesis. So these three stages are universal whether it is a prokaryotic system or the eukaryotic system. In the prokaryotic system, because the cells are very small and the system is very simple, we don't actually divide that into the different phases. But in the prokaryotic system also, you have the three different stages where you are actually going to have the synthesis of cytosol and the synthesis of the genomic DNA. Then you are going to have the division of the division and the distribution of the DNA between the two half of the cells, and then there will be a cell division by the cytokinesis. So these are the three stages. You are actually going to have the G1 phase, where the the cellular machinery is going to synthesize the different types of biomolecules which is required for the DNA synthesis. So you are going to synthesize the DNA polymerase, you are going to synthesize the nucleotides and all that. Once the cell is ready, then it is actually going to cross G1 to S phase, and then it will enter into the S phase, and within the S phase it is actually going to do the DNA synthesis. Once the DNA synthesis is over, then it is actually going to enter into the G2 phase, and G2 phase is actually going to synthesize all the machinery required for the cell division, and then it will actually going to enter into the mitosis, and then post mitosis, once the cell is going to divide the DNA between the two daughter cells, then it is actually going to perform the cytokinesis, so that the cell is going to divide into two parts. One is the mother cell and the other one is daughter cells. So during the cell interphase, you have the two different phases. One is called as the interphase, right? So within the interphase, you have the G1 as G2 phase, and then you have the M phase, which is the mitotic phase. So in the interphase is the preparatory phase required to perform the requisite step. These are the series of events in the nucleus and as well as the cytoplasm of the daughter cell to enable it to enter into the division phase. These phase has several other phases such as G1 phase. So it is also known as the growth phase, right? So it starts from the end of the mitosis and until the beginning of the S phase. This means during this phase, right? So it is actually going to start by the end of the M phase, and then it will end at the beginning of the S phase. During this phase, the cellular proteins, enzymes are synthesized. What are the enzymes are going to be synthesized? The enzymes are going to be synthesized which are going to participate into the S phase, such as DNA polymerases. It is going to synthesize the helicases. It is going to synthesize the topoisomerases. It is going to synthesize the single stranded DNA binding protein, gyrase, and all those kind of cellular machinery. All these machinery and their detailed discussion we are actually going to do when we are going to discuss about the central dogma of molecular biology, where we are actually going to discuss in detail about the DNA replications. So most of these enzymes are or the protein components are required for the DNA synthesis in S phase. Duration of the G1 phase depends on the cell type within the organism. So G1 phase is under the control of the P53 gene product, and remember that when we were discussing about the lecture number 1, we discussed about in detail about how the P53 is regulating the cell cycle and how the P53 activity is suppressing the tumor activity. So it is actually because the P53 is controlling the cell cycle in such a way that it is not allowing the the rapid duplication of the cells, and it is always wants the cells to go for the division phases under the tight control. Then once the G1 phase is over, then it is actually going to enter into the S phase, and the once the cell grow and all the factors, like what are these factors? The factors like enzyme, the factors like nucleotides, right? So are going to be available. It is start the DNA synthesis during the S phase, which means during the S phase you are going to have the replication of genomic DNA, right? At the end of this, all chromosome present in the nuclei is replicated and the DNA content is going to be doubled. So there will be no change in ploidy, which means the cell if the DNA is going to be duplicated, but there will be no change in the ploidy. The synthesis of the DNA occurs very fast to avoid the exposure of newly synthesized DNA to the mutation. And this is very, very important that when you are synthesizing the DNA, it has to be done in a discrete steps, right? It has to be done in a phase wise. And on the other hand, you are not going to open the complete DNA. For example, if you have a genomic DNA like this, right? Then this is just an example, right? Suppose you have this, right? So you are actually going to unwind this DNA, right? And then you are going to perform the replications. Then you are going to do the unbinding of this DNA and then you do the replications. Then you are going to unwind this DNA and you are going to replication. While you are doing the replication of this part is already going to be closed. This part is going to be closed. This part is going to be closed. And then you will enter into the next phase. So that a very small amount of DNA is going to be open and exposed for the environment, and that's how you are actually going to ensure that the synthesized DNA is few or free of the exposure to the mutagens. Then once the S phase is over, then it will enter into the G2 phase, the growth phase between the DNA synthesis and the mitosis. During this phase, the cell grow and synthesize the protein and the cellular factor required for the M phase and the cytokinesis. So once the G2 phase is over, then it will enter into the next phase, and the next phase is called as the M phase, right? M phase is actually distributing the DNA between the sister chromatids, right? So after the G2 phase, the cell enter into the M phase, and to divide the DNA equally between the two total cells, each mitosis has four distinct phases to precisely divide the cell. So during the M phase, you have the two options. One, you are going to do the mitosis, or you are actually going to do the meiosis, right? So mitosis is actually going to divide, and it will be going to distribute the DNA between the daughter cells, whereas the meiosis is also being called as the reduction duplication, right? So here the DNA is actually going to be further reduced. So you are actually going to reduce the ploidy of the cells, right? So if you started with 2n, it will enter, and after the meiosis, it is going to be n, so that all the cells are actually going to have the n ploidy. So we will discuss about the mitosis and the meiosis, and you will understand what is the significance of the mitosis and what is the significance of the meiosis. Apart from this, you also have the another phase which is called as the G0 phase. So after the G1 phase, some of the quiescent, senescent and non-proliferative multinuclear cells enter into the G0 phase. So G0 phase is a non-dividing phase, right? So many of the cells which are into the terminal stage, which are actually not been allowed to divide, and they are under the quiescent or senescent stage, they will enter into the G0 phase. Cells remain in this phase for long period or indefinite period, as in the case of neuron. It is also common in fully differentiated cells. The fast growing cells undergo into the G0 phase, and it is not a regular cycle, and under specific conditions cells enter into the G0 phase. So whatever the cells will enter into the G0 phase, they will not going to divide, right? Now how we are going to analyze all these stages, how you are going to analyze the G1 phase, S phase and G2 phase and the M phase, right? You can actually be able to use a DNA binding dye. Remember that the amount of DNA present throughout this cell cycle is going to vary, and that can be mapped with the help of the DNA binding dye. So one of the classical DNA binding dye is called as propydium iodide. It is a fluorescent dye, and this is the structure of the propydium iodide. What you see here is the actually having the positive ion onto the dye, and it is also going to have the two iodine molecules, and it is being used for the analysis of the cell cycle to label the DNA. So what you do is you label the DNA within the cell, and the cell is actually going to have the different amount of DNA during the G1 to M phase. PI has the unique property of being unable to enter the live cells, but capable of permeating the cell membrane of the decreased or the dying cells. Through its binding of the DNA and subsequent emission of the red fluorescence upon excitation with a specific wavelength, PI enables the evaluation of individual cells, the DNA content, and facilitate the examination of their distribution within the cell cycle. What are the material you required for performing the cell cycle analysis by the PI? So you require the 6-well plate, you require the propydium iodide, you require the RNAs, you require the PBS, the alcohol, flow cytometer, centrifuge, 15 ml Falcons, 1.5 ml centrifuge tubes, trypsin EDTA, and the 37 incubator. How you are going to do the cell cycle analysis? So what you do is you seed 10 to power 6 cells per well in a 6-well dish 12 hours prior to experiment. So this means you can probably do in the night the seeding of the cells, so that in the morning you can come and do the treatment. On the day of experiment, wash the cell twice with PBS and treat the cell with desired concentration and incubate for 24 hours. This timing is not very strict. You can do 12 hours, you can do 18 hours, you can do 24 hours, you can do whatever the time, right? It depends on the treatments and other kinds of parameters. After 24 hours of treatment, remove the media and scrap the cells with the trypsin or the 0.6% EDTA, depending upon what you want to do. So if you use the trypsin, the cells are actually going to come off by cleaving the cell adhesion receptors, but if you use the 0.6% EDTA, it is not going to do that. So depending upon your overall objective, you can use the trypsin or the EDTA. Then you centrifuge at low RPM and discard the supernatant so that you can collect the cells for your analysis. Then you do the staining of these cells, so you can keep the cells in the 250 microliter of PBS and keep the suspension on ice for a couple of minutes. Then you add the 1 ml of 70% ethanol and you fix the cells. Then you transfer the cells into a minus 20 degrees Celsius for 3 to 4 hours. After incubation with 70% ethanol, centrifuge the sample at 5000 RPM for 10 minutes, discard the supernatant. Then you add the 1 ml of ice cooled PBS, centrifuge the sample at 5000 RPM. And after the RNA degradation by the RNase, you stain the cells with PI, right? The final working concentration is 50 to 70 micrograms for 30 minutes before acquiring the data in a flow cytometer. After that, you are going to data acquisition, so you are going to use the flow cytometer, and so after staining, analyze the cells under a flow cytometer equipped with the appropriate filter for the propylium iodide. So propylium iodide has an excitation wavelength of 488 nanometer and an emission wavelength of 585 nanometer, which means you probably can use the appropriate excitation and emission filters and adjust the flow cytometer setting for appropriate fluorescence and forward and side scattering parameters. So this is all we are not going to discuss in this particular course, but if you are interested, we have discussed about these instruments in my another MOOCs course, which is called as the experimental biotechnology. So where you can actually get the more detail about how we can be able to use the flow cytometry. So you can actually go through with that lecture, right? So it's called experimental biotechnology. Run the stain sample on the flow cytometer, collect the data at least for the 10,000 events. Analyze the acquired data using the flow cytometry software. Plot the DNA content, which is actually going to represent the signal from the propylium iodide, and SL count on the y-axis. The resulting histogram will typically show the distinct peak representing the different phases of the cell cycle, which means the G1 phase, S phase, and G2M phase. And then you can analyze the data using the analysis software, which is called as the FCS Express software. While you're doing these experiments, you are supposed to take a lot of precautions. So to avoid any disruption in the distributional cell population, it is recommended not to seed or treat the mammalian cell when they reach a confluency level, which is the 90% to 100%, which means if the cells are very old, right? If you are taking the cells of 90% to 100%, they are going to be very old, and then they may not be actually responding to, or they may not be show the distribution of the different cell stages. This is because at this stage, the majority of cells are in G1 phase, and using the cells that have attained 100% confluency can impact the overall distribution of the cellular population. To avoid obtaining unwanted background signal, it is important to run the cell labeled with the propylium iodide in the PBS when using the flow cytometer instead of using the complete or the incomplete medium. And then you need to ensure the excessive pipetting when scraping the cells from the plate, because that is also going to damage the cells, right? So we have prepared a small demo so that you can be able to understand how you can be able to use the flow cytometry and how you can be able to use the propylium iodide to study the cell cycle analysis.
Hello everyone. In this video, we will be discussing about how to perform cell cycle analysis on a fax equipment. So basically, the cell cycle analysis gives us the details about how much population of a given number of cells is present in which phase of the cell cycle. For example, the G0 or the G1 phase, the synthesis phase, or the G2 or M phase. So we can design the experiment according to our needs. For example, if we want to find out how a chemical compound inhibits certain population of cells, we can do a time dependent experiments, or also we can do a concentration dependent experiment. So in both the ways we can find out which particular phase of the cycle is being inhibited. The procedure for cell cycle analysis is very simple. First, we trypsinize the cells from 100 mm cell culture dish and seed approximately 1 million cells in a 6-well plate. After the cells are seeded, we incubate it for 12 to 14 hours so that the cells get adhered completely to the dish. After the cells are adhered, we give the appropriate treatment, but after washing it twice with PBS. So after the appropriate amount of treatment, let's say 12 hours or 24 hours or 48 hours, we trypsinize the cells and collect the pellet. We wash the pellet with 2 times with PBS and then centrifuge again, and then resuspend the pellet in 1 ml of 70% ethanol. This mixture is kept in -20 degrees for 12 hours for fixating the cells. After the cells are fixed with 70% ethanol, we wash the cells with 5 ml of PBS and then collect the pellet and resuspend the pellet in 1 ml of PBS. Then we provide the appropriate RNase treatment to remove all the RNA from the mixture, because RNA, if present in the mixture, might interfere with our cell cycle analysis. After incubating the sample with RNase for 2 hours, we give the appropriate propydium iodide treatment. The working concentration for propydium iodide in cell cycle analysis is 50 microgram per ml. After giving the treatment with propydium iodide for around 1 hour, we analyze the cells in FACS equipment. After the samples are prepared, we need to analyze the data in the FACS equipment. The first thing we do is open the Cell Quest Pro software. After that, we connect it to the cytometer. After connecting to the cytometer, we need few things such as counters, detector and amps, status, and also we need the dot plot. Press acquisition and analysis. We need another dot plot for FL2A and SSC, because in the FL2A channel the propydium iodide emits red fluorescence. We also need a histogram to see the cell cycle phases. For that purpose, we need the FL2A channel. We need another dot plot in order to see whether there are presence of any doublets. There is a chance that there might be a presence of the doublets in the data, like for example the clumps of cells, so we have to exclude that from the data. For that purpose, we take the FL2A and FL2W channel. Now that everything is ready, now we can change the directory for acquiring the data. We can also choose the file name, so we'll name it as untreated1 and we'll keep the file count to 1. For checking the data, we have kept it on the setup so that we can see whether the data is coming properly or not, and then we can remove it from the setup and then acquire the data. So now we are going to load the sample onto the sample injection port and then press acquire. As we can see that the number of events have started recording. In the FSC and the SSC plot, we can see that most of the population is seen between the 200 and the 200 mark. The population which is coming away from the doublets in FL2A and SSC, we can see that there are 3 different populations, G1, S and G2. And in the FL2A and the counts, which you can see that the longest peak is the G1, the between one is the S, and the smallest peak is the G2. So before acquiring the data, we have to set the number of events. So we go to acquisition and storage and press record 10,000 events. So in order to record the data, we have to remove it from the setup and then press acquire. In the FL2 and the FL2W channel, we can see that there are 2 different populations. One is a thin line and other is the presence of some doublet or clumps of cells. This distinct population might be presence of the doublet cells or clumps of cells. So while acquiring the data, we can see this, but in the FCS Express software, we can remove that. So now that we have acquired the data, now we can take the data from the treated sample. We have to remember that we don't have to change the parameter conditions in order to check the compare the untreated and the treated samples. Now we have to change the sample name from untreated to treated, and also change the file count to 1. It's okay, and then acquire. We can see that there is a little shift in the S and the G2 phase, but we can't say for sure that there is a change in the data. So for that we have to process the data on the FCS Express software in order to see what is the difference between the untreated and the treated sample. So now we will remove it from the setup and then acquire the data for the treated sample. Sometimes we can see that the events per second will be low. So in order to increase the events per second, we can pause the recording and then tap the sample once or twice in a while to shake the sample, and then the flow will be continuous again from the sample injection port, and the events per second will increase. Now we can see that the events per second has increased because we have tapped the sample, and the data acquisition will be a little bit faster. So now we have acquired the data for the untreated and the treated samples. In order to see in which phase of the cell cycle the arrest has taken place, so we have to process the data in the FCS Express 5 software. So now that we have acquired the data on the fax equipment, we have to now process the data in order to see the difference between the untreated and the treated sample. For that purpose we use the FCS Express 5 flow software in order to process the data. So for that purpose we use the new layout and then change the mode to landscape because it is easier to work in the landscape. And then after that we go to the data in the toolbar and press open, and then go to the folder where our data is saved and press open the untreated file. As we can see that there are multiple options available: dot, density, color, dot, contour, surface, histogram, and multicycle DNA and kinetics. For this cell cycle DNA we only need multicycle DNA plot and the dot plot, so we are going to open these two. Because we have recorded our data on the FL2A channel, and also because the propydium iodide is only shown in the FL2A channel, we are going to open the FL2A channel. So now after we have opened the dot plot and the cell cycle DNA plot, we can see the how much population of cells is represented by the G1S and G2 by right clicking on the plot and then selecting statistics and then show DNA cycle statistics. So a small window will appear in which it will show that the percentage G1, the percentage G2 and the percentage S. So this is for the untreated sample. Let's see for the treated sample. As we can see that there is some change in the untreated and the treated sample, both in the both plots, the dot plot and as well as the cell cycle plot. So in order to see how much percentage of the cell cycle phases have changed, we right click on the plot, then show DNA cycle statistics, and then place it right beside the untreated one in order to compare. So we can see that there has been a reduction in the G1 phase from 61 to 49, and from G2 also from 15 to 12, but there has been an increase in the percentage S phase, which has increased from 23 to 38. So we can say that there is a significant change in the phases of the cell cycle, but we can only be sure after doing the experiment in the triplicate, so that we get the proper standard deviation and also the standard error. So this is one way of processing the data. The another way of processing data is by gating. As we can see in the FSC and the SSC channel, that there are some debris near the 00 point, and also there are some population of cells which are very far from the 00, representing clumps of cell or maybe doublets, which we have to take care using gating technique. In order to explain the gating, I will use another page and then open the data and then press OK. So for gating we need multiple plots. The first one is FSC and SSC, and the second one is between FL2A and SSC, and the third one is between FL2A and FL2W. The fourth one is between FL2A and FL2H, and the last one is between FL2H and FL2W. So we have to start the gating from the first plot, the FSC and the SSC. So in order to do the gating, we have to go to the gating option in the toolbar, and then we can choose any one, like ellipse or the rectangle, polygon, freeform. These are the shapes of the gating. So we'll go with the polygon gating because it is easier to handle. And then we can gate our required population using polygon gating tool, and then just select the required population. So here we are excluding the debris and the like the clumps of cells which might be interfering with our actual data. And then we can select what gate color we can give, and also rename the gate 1 to our particular type, but we'll go with the standard one, and then press OK. And then we have to apply this gating to the second plot. We can just drag and drop it on the second plot. So see, we can see here that we have excluded some population. So but still there is some population which can still interfere with our data. So we'll go do the gating again, and then we'll exclude some more population which might not be helpful to us, and then we'll name this as gate 2. So in order to apply this gate to the third plot, just drag it and drop, and then we can see that another number of population have reduced, and in the third plot as well we can reduce this population which is a little bit distinct from the singlet cells. So we have taken another gating using polygon, and then we are only now going to select the singlet cells, and then we'll name this as gate 3. From gate 3 onwards, we just have to apply the gates to the next plot, and then we can apply the gate to the plots using by just selecting the plot and then going to the top left corner and then selecting the gate. So as we have seen that we have reduced a significant number of population, and we have only selected the population which might be helpful to process the data. And then finally we apply the gate 3, which is the final gate, as then you will see the change in the cell cycle statistic plot. So this is for the untreated one. In order to show the cell cycle statistics, just press statistics and then show DNA cycle statistics. As we can see in the ungated one, we have seen that the percentage of G1 was 61, whereas in the gated one it is 67.1, and the percentage G2 is 8.9, and the percentage S is 23. Similarly, we can do for the treated one, but we don't have to follow the whole procedure. We can just copy all the plots to a new page, and then press paste, and then go to data, and then select the treated one. It will just replace all the plots with the treated data. So but the treated data and the untreated data is little bit different, so we just have to move the gate. So in order to see the cell cycle statistics for the treated one, we can select the statistics, DNA cycle statistics, and then we get the DNA cycle statistics. So in this way we can process the data for the untreated and the treated sample in the cell cycle DNA statistics using gating as well as non-gating technique. So hopefully this video was helpful for everyone.
Once you analyze and you get the data, right? So this is the result what you are going to get, right? So this is the sample from the untreated sample. This is a cells treated with the anti-cancer compound, right? So for example, this is actually the data from the NDA MB231, the breast cancer cells, and they are either being not treated or the treated with the anti-cancer compound. So what you see here is the this is actually the G1, right? So this is the G1 phase, this is the the G2 phase, right? And this is actually being called as the S phase, and what you see here is very small, which is actually going to be called as the M phase, right? So what you see here is a lot of cells are there in the G1 phase, and then there are cells also into the G2 phase. As and then you will see if the percentage wise, if you see the 54% cells are in G1 phase, 67% in the G2 phase, and 39.4% in the S phase. When we treated it with the anti-cancer compound, the cells present in the G1 phase is 72. The cell present in the G2 is also 6.41, but the cell present in the S phase actually got reduced and it becomes the 20%. So that means it is actually affecting the cell cycle, the treatment, whatever the compound you are using, it is actually affecting the distribution of the cells within the cell cycle. So, what is the role of the cell cycle? So, we have discussed about the cell cycle, we have discussed about the different stages of cell cycle, but what is the significance of following the cell cycle? So, it is required for the development and the growth, it is required for the cell replacement, it is required for the regenerations, it is required for the asexual reproductions. So, talking about the development and growth, the development of a single cell into a multicellular system is possible due to the cell cycle and cell divisions. And talking about cell replacement, the eukaryotic cells have a predefined lifespans, right? And after the lifespan is over, then it has to be replaced, right? So, and after that period, it needs to be replaced with a new one, and it is possible due to the cell division and making more cellular copies. For example, the human RBC has a lifespan of 3 months or approximately 100 days. New RBCs are formed from the bone marrow by the cell division. Similarly, we have the regeneration. So, cellular damage and injury is the integral part of the living system. Cell division is the primary event required for the synthesis of the lost or the damaged organs. So, it could be an injury from inside or it could be an injury from outside. So, when you get a cut, right, the blood comes out, right, and then the regeneration process starts, right. Similarly, you might have seen the regeneration into the lizards and all other small organism where they actually be able to regenerate the whole organ, and in this process the mitosis is actually taking place. Then it also be required for asexual reproduction. So asexual reproduction is common in lower invertebrates. In these organisms, the cells divide to form the new cells, and these newly formed cells give rise to the new organism. For example, the hydra, amoeba, and all those kind of new organisms. Now, the cell cycle has to be under tight control, and that is what we have discussed in the lecture number 1, that how it is different. There is a complete set of protein machinery which is doing, recognizing the different stages of the cell cycle, and that's how they are regulating the cell stages, right? So, cell cycle at a different step is tightly regulated by the cell cycle checkpoints, and these cell cycle checkpoints are used to ensure the completion of different steps and repair of the cell damage. The main checkpoints are present at G1 to S, G2M and M phases, and each point is controlled by the mutual interaction between the cyclin and the CDKs. And this all we have discussed in detail how the regulation is happening. And the P53 and RB protein, all these are also being involved into the control of the cell cycle stages with the G1 and G2M. Now, the question is, what will happen if these events are going to be wrong? So, dysregulation of the cell cycle and control mechanism give rise to the tumor. After certain number of cell division, every cells enter into the G0 phase and ceases the cell division. In the case of tumors, cells lost the control and multiply indefinitely to give rise to the cell mass, and these cells are taking nutrition but not performing the function. So, retinoblastoma, like the RB proteins, P53 are the crucial cellular factor responsible for the cell cycle control and play crucial role into the tumor development. So, we have discussed in detail about the cell cycle, we discussed about the cell division, we discussed about the growth media, we discussed about the different stages of the cell cycle and so on. If you are interested to study more, and if you want to study more about these events, then you can actually be able to follow this particular research article, and this research article is very good in terms of explaining you the role of the checkpoints and the role of cyclin and the CDKs. So with this small discussion about the cell cycle and how you can be able to study the cell cycle into the different into the mammalian system, I would like to conclude my lecture here. In our subsequent lecture, we will discuss some more aspects related to the cell division and cell cycle. Thank you.